I called this first core lecture embodied cognition, even though I will only be talking about embodiment at the end of this lecture, but you'll see it's sort of the founding idea of DFT is this binding or link to the sensory and motor surfaces. So welcome to our first core lecture, okay? So I have this feature in these lectures that I have this little blue marker up here, which gives you sort of a table of contents of where we're going next, sort of to orient a little bit. I hope that works. So for instance, now I'm announcing with this roadmap slide that I will be talking about neurophysics first, about spiking and integrating fire neurons, and then about attractor dynamics, and then point in the end, we'll point to the fact that such dynamics are just low-pass filters of input. Maybe Jan can respond to the question about recordings as well. So here we go. And so this is from a textbook, actually a very nice textbook, Brazilian, and illustration just of a recording, in this case, of an axon. When you take a neuron, you inject some current into the neuron and record somewhere downstream from there, then you will see observe the membrane potential. You may know that the membrane potential of neurons is always negative, and when it goes toward the positive, then that's excitation. So what happens if you insert these little currents, that the system responds to the onset of the current by growing, and it's growing in an exponential form that can be well understood as being the result of a linear dynamical system of this nature. If you use the activation, and if you turn it off, it relaxes in an exponential form again. There's actually timescale is slightly different in the two cases, but we can neglect that. And the amount of response depends on the amplitude of your input. More response for more input, and at some point something different happens, a spiking, which I'll come to in a moment. And if you look at the timescale, this is a caricature of real neural data. You see here, this is milliseconds. You can almost sort of guess that the timescale on which the membrane really of physics, neurophysics, the biomechanical, or it's a biochemical system with electrical charge and ions and so on, that timescale is about 10 milliseconds. And it will turn out that we will be looking at neural dynamics at the population level at the same time scale. So this factor tau that determines the relaxation time of the exponential time courses is about 10 milliseconds. If you don't understand equations like that, I'll explain those in a moment in a more accessible form. So right now, I'm just assuming you know such things. So if the induced activation level exceeds a threshold, there's actually the spike event, which I'm not explaining how that happens, but that is really what is transmitted. So when the axon connects to other neurons through synapses, they only induce some post-synaptic potential in the downstream neuron if there is a spike. If the spike is the self-generated, sort of normed, positive potential event. And you see there's a certain degree to which spike events need to overlap so that the different activation changes superpose enough to reach threshold. And that's very important for spiking neural networks. The rate at which a neuron spikes, it reflects the level of input. So if you have a low level of input, for instance, you may get a low rate. If you have a higher level of input, you might get a higher rate. Still sort of shockingly low, these rates, right? Typical rates would be 10, 20, 30 hertz. So that would still corresponds to having interspike intervals. At 100 hertz, you would have an interspike interval of 10 milliseconds. So that's not very dense firing. So that's always something to be aware of. Now, in our modeling, just like in all of connectionist modeling, we don't really deal with the detailed membrane and spiking and synaptic mechanisms. We have a simplified description for those. So we replace the spiking mechanism itself by a threshold function. So a threshold function essentially says that if activation is sufficiently positive, we conventionally just put the threshold at 0 of some kind of more abstract activation variable. Then the response that will be transmitted to downstream neurons starts to deviate from 0, ultimately being 1, some kind of normalization. And that's a description that has been derived as a description of what populations of neurons do. And there's a lot of theoretical work on exactly what that, under which conditions you can derive a description like that. But that is the level of descriptions that we'll be using. And if you do that, then you recognize that the activation variables we're using are a little bit more abstract than these physical membrane potentials. First of all, we give them a sign, so they go from negative to positive, and we set the threshold at 0. And ultimately, what are the units? They're not really in millivolt. The units are really relative to some kind of sigmoid. If you choose a certain threshold of that sigmoid, then that really determines a certain steepness of that sigmoid. That really determines what range of activation matters. So for instance, you have a very steep sigmoid, then only activation that lies within this little interval makes a difference. If you have a broader sigmoid, then activation changes in a broader range make a difference. So it's ultimately a question of downstream neurons, so in a neural network, the neurons onto which a particular subset of neurons projects, that determines the meaning of activation. Ultimately, that will be the motor system. And today's lecture will go all the way to the motor system and sort of understand what happens there. But we'll do that again in somewhat abstract terms, not really dealing with actual muscles, for instance. So activation is a bit more abstract, but that's the same abstraction that is done in connectionist networks. So many of you will be familiar with that. Now, unlike connectionist networks, we'll consider these neural activation variables to be dynamical variables, and that means they are described by differential equations like that, right? So abstractly, so differential equations are one concrete instance of what abstractly is called a dynamical system. You can say a dynamical system is very generally a system in which there is a description by some state variables, we call these here activation states, such that given that state, you can predict the future. You don't need to know the whole history. You don't need to keep track of time into the future. You can just take any instantaneous state and predict the future. So technically speaking, that means it's the existence and uniqueness theorem that if you have an initial level of activation at some point, arbitrarily called 0 here, then the time course of activation for larger times is uniquely determined by that initial condition. So you might need multiple activation variables if you have a whole network, but it's always just at one given initial moment in time that you need the activation to predict the future. If you think of stochastic processes, this corresponds to the Markov property. Markov property says that you need the probability at a particular point in time, and the dynamical rule is that transition probability that corresponds to the differential equation, if you wish. And then the future can be computed. You don't have to keep adding new information. You can just generate it from that dynamical law, which is the transition law. So the form of there are multiple mathematical instantiations of dynamical systems. For instance, some famous versions use discrete time, like dynamical maps. I'll not do that here. It's not a good description of the nervous system, in spite of that spiking, but these spikes are actually asynchronous. It's not like there's a clock that updates the whole network at one point. So it's not a good description to use discrete time, even though some some connectionist models use that, but it's more like as a simplification. So time is actually continuous, because the spikes can happen at any moment in time. It's a continuous system. You see that in the membrane dynamics, which is this integrate and fire description, and that's time continuous. And in time continuous form, dynamic systems have this form of a differential equation. So that really means that the rate of change of one variable, the dot here is the rate of change, so the derivative with respect to time, is a function of the current state. So you always plot the rate of change as a function of activation here for a single variable. We call that then the dynamics, the dynamical system. So rate of change, just think if activation was distance, then the rate of change would be speed, right? It's the change of distance, that's the speed with which you move, velocity. And so it's the activation velocity, if you want, the rate at which activation changes. So very general is this form where we have a minus u term here. So the minus u term is a negative slope. And what that does, so that's specific, that there could be any function, and this is a specific function. And I showed previously these examples that here, that this negative function here is the right model to explain these exponential time courses that result from stimulation of the membrane. And that is a postulate for our kind of models, and it has a deep meaning. It's ultimately radiates into the entire theory, and I'll illustrate that here around this problem. So when you have that negative slope, what you get is that a straight line like that will intersect at some point the u-axis. Intersection like that means that the rate of change at that point is 0. And we call that a fixed point. And a fixed point like that is a solution of the dynamic system. It's a constant solution, right? If you have that level of activation, your rate of change is 0. So you're going to stay at that level of activation. So time constant, stationary solution. It's you can compute that stationary, fixed point solution by just setting the derivative equal to 0. If you set that equal to 0, put this onto the other side, then you get that the fixed point is at this number h. We conventionally make this number h a negative number. That's why it's here to the left, because we're considering the threshold to be 0. So something below 0, because we're assuming that in resting, without any other inputs that we'll come to in a moment, nothing is transmitted downstream. No motor system is driven, so on and so on. So better be negative. Now, the slope of that dynamical function is negative. We have a minus here, and that creates stability. So this is called a fixed point attractor or stable fixed point, which mathematically means that if you're starting, initial conditions start nearby, here actually it can be anywhere on the u-axis, but in general it's just nearby, that you then converge in time to the fixed point. And that makes it stable in the engineering sense. That is, if some disturbance pushes you away from that fixed point, then you return to the fixed point by that convergence feature. The negative slope predicts that. It's very intuitive. If you start here at a higher activation, you have a negative rate of change, right? So your next activation level would be to the left, and look at that up again to the left and so on. So you would be, if you sort of make a mental iteration as a mental simulation of the system, you see that you would be approaching the fixed point. And if you're there, you don't change anymore. It will be fixed, right? Similarly, if you start to the left, you have a positive rate of change. So your next moment in time, you would have a larger activation. Again, you could iterate that in your mind. You see that you will be converging to the fixed point. So this attraction comes from the negative slope. And that's a very general theorem in higher dimensions. It's the negative real part of the eigenvalue of the underlying linear dynamics that makes that stability. Now, this just summarizes what I just said. That is, if you start from different activation levels, you will be converging in time to the fixed point level, which is here just the resting level. And you see there's some kind of contraction. This is an example of what is called the flow in dynamic systems thinking. That is a survey over the ensemble of all solutions, if you think of all possible initial conditions. And mathematicians actually like to look at dynamical systems that way. They call these differentiable diffeomorphisms, where they think of the set of all initial conditions and then how they are mapped as a function of time to new states. And so this will be a converging flow. And that is sort of illustrates that attractor solutions are really very interesting because they characterize the ensemble of the solutions. They characterize what happens for any initial condition. So even if in a dynamic system, as we'll later see, you never reach the attractor, because before you've reached the attractor, something changes again and you go somewhere else. But you can still say the attractor sort of reflects the organization of the system. So, qualitative features of the ensemble of solutions. And that's one reason why knowing about attractors is very important. It characterizes the ensemble, the flow. Now, that's just without input. That's just a single neuron doing that. Once you add input, the dynamics is going to be changed. So here I'm thinking of input as a contribution on the right-hand side, a contribution to the rate of change. So for instance, a positive contribution would be called excitatory input. A negative contribution would be called inhibitory input, and would come in the membrane dynamics. It comes from the postsynaptic potentials of the corresponding synapse. The membrane is the same all the way up to the dendrite, and just past the synapse, you would have a negative or positive postsynaptic potential. That would be the source of input in this neurophysics example. So it's important to realize that inputs are now really contributions to the rate of change. They're not direct. You're not directly setting the activation. So unlike some feed-forward neural networks that connectionist networks that sort of neglect that, they would just compute the output as a function of the input. But if you take into account the actual dynamics of the membrane, then you get get that it's a contribution to the rate of change. So for instance, if in this previous example that I had, this was your dynamics in the absence of any input, you would have that resting state as an attractor. If I add an input, it's a constant contribution, constant in the sense it doesn't depend on you, right? Might depend on time, so it's not autonomous as we would say mathematically. It could be time dependent, but it doesn't depend on you. So in other words, the whole curve across all you is just lifted up, and so this black line here is just the gray curve lifted up by a certain amount. And you see, as a result, the attractor shifts. It looks like the attractor shifts here to the right, but it's really brought up by the curve being lifted up vertically. And so what happens is if you're sitting here in this attractor, suddenly this input arises, like in this example, would arise instantaneously, let's say, then your rate of change jumps from 0 to some very positive rate of change, and then you would be running to the new attractor. And so you're running to the new attractor that's now in the positive range here as a result of that minus U term, of that stabilization term, right? So here, this is an illustration that the simulation, step change of the input, the actual activation level, relaxes exponentially to a new level. That will be this H plus S. U is not equal to S because it's lowered by the resting level. This H is added here, right? If you figure that out, U dot equals 0, put U to the other side, then the new attractor is H plus S. Of course, if S is time changing, the attractor would be moving in time. It's no longer strictly speaking an attractor. It would have to be moved slowly enough for that to be still a good parameter. I've plotted in here the sigmoid of that. You see the sigmoid, if it's steep enough, would start deviating from 0 only once activation goes through threshold, so somewhere here, and would saturate at a particular level. Good. So what's transmitted ultimately is that Cian curve here, the sigmoided, we sometimes say sigmoided, which is not a really English word, activation. So that's very important to understand for the rest of this lecture. So in a way, this is still sort of trivial, right? In a way, all that happens here is that a time-varying input is, we say, low-pass filter. That is, where if the input jumps, then the solution smooths that jump. That's the only contribution in the sense of the dynamics up to now. That's certainly something that the neurons do, right? I showed you how the neurons actually create these little exponential transients. But we'll see that that has a lot of other consequences in a moment. The last point I want to make about these individual neurons is that you see that the minus U term is special. So what will happen in neural network is that we'll be adding all kinds of inputs here, right? A neuron could integrate a lot of inputs, have a sum of inputs. And so positive inputs will be excitatory, negative inhibitory, and you think of the connectivity being reflected in that. While the minus U term changes sign, right? It could be positive or negative, depending on the sign of u. So that's not explainable as a driven by some input, or doesn't reflect any synaptic process. It is essentially, in the neurophysics example, it's an intrinsic property of the individual neuron. And it is the mechanism that makes stability. It's the source of stability for the whole system because of that negative slope. So the minus u term is special, and it's sort of often unexplained if you look through, for instance, some of Stephen Grossberg's paper, who was sort of the pioneer of using neurodynamics. And it's not always so clear where that comes from. Yeah, and it's ultimately the membrane. It's sort of the neurophysics that it comes from. I mean, it's clear to him. It's just not always expressed because it's maybe taken for granted. Okay, I thought I even do a little simulation with our simulator for that. It's kind of trivial, but I do that. So this is the 2-neuron simulator that is on that webpage that you can play with later. There are 2 neurons here. So activation 1 is up here, node U1, it's activation dynamics, and then node U2. We need that later. They are plotted against each other here on the left. This is a running time course of the activation. This is the rate of change U dot against the activation itself. The red curve is that dynamics. Here's the fixed point. Currently, it's at -5. And so the system is sitting in the fixed point. That's why it's here at -5. But if I add some input to the system, I can shift this around. And as you see, what happens is the system low-pass filters that input, right? If I shift this more or less abruptly, it's a question of how quickly I move the slider. Then you see that the system just exponentially relaxes to a new level. And that is what the low-pass function is. And so here on the right, you see, in terms of the dynamics, it just means that I'm shifting the dynamics up and down. And you see, it's always the -u term that makes that the system returns to the attractor. So if you look with the naked eye, it looks like it's moving fast when I'm shifted a lot, and then it slows down, which is really true because of that -u term. The rate of change is large when I shift it a lot, but it has a constant rate. So the exponential function is that function that where the decay in a certain percent of distance is always the same. And that's why we can say an exponential function has a particular rate. I don't want to go too deep into that, but so take for granted that you know these sort of things. If not, that is actually discussed in the first book chapter, so you can read that a little bit. Good. So next step would be to now think of multiple neurons and multiple inputs, right? In connectionism, the way connection describes neurons is that there's some activation variable that ultimately determines the output, and that activation variable is directly set to be the sum of the inputs, and then the sigmoid generates the output, right? That's a connections network for you. So a forward connection network actually is a functional description where you're just saying the output is a function of the inputs, not a dynamic of description. And you could see that's sort of the case where you're neglecting that low-pass filtering, where you're saying, well, the difference between these time courses, I don't care about. And so I can just set the time courses to be immediately the time courses of the inputs. That will be the limit in which you arrive at that. And I'll show you in a moment that when you look at recurrency, then the time courses are really necessary. And that approximation breaks down. Now, before I go there, I want to talk about neural fields already. So neural fields just as the creating many neurons and then essentially determining what these neurons mean by how they are connected, ultimately to where they get input from or where they send their output to. And so here the roadmap for this next couple of slides is to look at patterns of forward connectivity from and to sensory motor surfaces, tuning curves, receptive fields, analogous to forward neural networks, and see how then when we sort of smear out neurons and no longer consider them discretely separate, we get to neural fields. and then briefly talk about what feature spaces then mean. So, so this is this connections picture I already showed, and a concrete example, a couple units that get input, and then they project, it's a forward neural network, that project that input further. So ultimately, the sigmoided output here of the last unit is a function of these inputs here. So if you have a network like that, people talk about neural activation patterns standing for something, so encoding something, right? A code is really just a invertible function. Now, if Morse code, it means letters are represented by something. So the dot pattern, let's say dot dot dot, stands for a certain letter. So it's a mapping from a letter to this, alignment of short and long signals. That's Morse code. And so people talk about neural activation in terms of coding by thinking of something outside the nervous system, like a stimulus or a motor task being the input, and then you're creating a function that's the inner representation of that. And so there are two versions of coding that are often invoked. One is called space code or labeled line code. And space code would mean that what a neural activation means or stands for is determined by where it is in the network, by its spatial location in the network. So it's actually ultimately by the network, of course, by the position of the neuron in the connectivity. So this neuron stands for something different than that neuron because this neuron gets these two kind of inputs. So it stands maybe for some combination of S1 and S2, while that neuron stands for some combination of S2 and S3. That would be sort of the idea. Well, in rate code, it's the activation itself that stands for something. So that's something that you often find in the periphery of the nervous system. So for instance, you have some sensor that is sensitive to some physical thing, like a photoreceptor in the retina would be sensitive to luminance, and the activation, for instance, the firing rate of a photoreceptor in the retina would fire as a function of that intensity. It's typically then not a linear, but a monotonic function. So higher intensity, for instance, could under some circumstances lead to higher activation. Actually, photoreceptors is the other way around. It's monotonically negative, that works just as well. And that's a code in the sense that there's a invertible function from intensity to activation. So if you know activation, you can estimate what the intensity must have been. And you could similarly think of a motor neuron, for instance, encoding force generation by the muscle, because there's a certain amount of activation will generate that amount of muscle activation. So a similar functional relationship between a motor degree of freedom and an activation variable. Now, that's the theoretical discrimination. Generic neural networks don't even talk about this very clearly. And what that means is that they combine both codes. So very often the firing of a neuron depends on the intensity of these inputs, but it also differs in different locations of the network. So it also depends on where you are in the network, right? For us, that you will see makes a big difference, because this rate code is actually not very important once you are within our neurodynamic architecture somewhere in the center. They only plays a role in the periphery. So I want to make this a little bit more explicit and link it to the concept of a tuning curve or receptive field. So let's say we have a whole ensemble of sources of input. I call this a sensory surface. You can think of it as the retina, let's say, for the moment. And so I would present, so far I'm assuming they're discrete units, but later I'll smear that out and just assume it doesn't matter the sampling with the discrete units. So let's say the different locations here along the retina, and from each of those locations, you're driving different neurons. Let's say this could be in visual cortex, for instance, or through maybe some via stations, some interneurons in LGN or wherever. And these different neurons receive input from that surface, and that input has a particular pattern. I'm suggesting here that from every photoreceptor, there are some synapses onto a whole set of target neurons, but with a limited spread, so that, let's say, a target unit like this gets input from these three locations, and let's say a sensor like that spreads its input to these three locations in the network. That's sort of the standard picture of how the forward neural networks would be generating tuning. And so tuning would be observed when you're recording from one of those hypothetical neurons and varying the input and varying, let's say, input by stimulating only this receptor or only that or only that, and you would get different amount of responses at this neuron, and these different amount of responses reflect that the synaptic strength from this sensor to the neuron is larger than the synaptic strength from this sensor to this neuron, and then maybe there's even no connection anymore if you go further away, like this sensor doesn't actually drive that neuron at all. And empirically, you would be observing that as, for instance, the function of spike rate as a function of some feature dimension, like retinal location, that would make the receptive field profile, or if you're varying the orientation of some stimulus, then you would get orientation tuning. And I'll show you a concrete example from Huber and Wiesel for that tomorrow, actually, to talk about how multiple dimensions are combined. So if you take that picture, then you could generalize that and you could say, well, just any pattern of sensory stimulation will translate into a corresponding pattern among those neurons that have these kind of forward mappings. And that is what is actually called a map, from one population of neurons to another. Sensory neurons also neurons, and to some other set of neurons. And you could do that along the path, maybe from LGN to cortex and so on, from one cortical area to another cortical area. And it's the connectivity pattern in each case that determines what the map means, and would be the distribution of activation over a population that then is mapped onto a different population distribution across another, in another area. And we consider these maps to be fields just by neglecting the discrete sampling. This is historically was motivated by the fact that cortex consists of these layered structures, and that if you look at the connectivity along these layered structures, it's very homogeneous. So you see the same kind of dendritic arborization and very similar synaptic distribution as you move along these layers. So that and they strongly overlap. So if you look at neighboring neurons, their dendritic trees overlap very strongly. So you could think of these, they sample something, but they smear out, because of this overlap, they smear out what they represent, that one can actually neglect that discrete sampling. And there's no single psychophysical fact where you notice the discreteness of individual neurons. There are psychophysical effects where you get close to noticing the spiking, where you see some kind of binary digitization. You see just one photon, for instance, from one spike. Very extreme cases, but not really with respect to the discrete sampling of neurons, and for reasons that are deeply understood that I don't want to go into. In fact, you can make a population code with just 2 neurons, but I don't want to go into the more technical versions of that. So that's where fields come from. So when I talk about an activation field, I'm talking about a pattern of activation over a space, and I'm describing that space as a function of some other space that another population is about in an architecture, or some sensory space, or then also some motor space, and mapping this dimension onto that dimension without keeping track of the exact connectivity pattern. There will be a strong reduction in number of parameters, because if I do this mapping, I characterize that mapping by a few parameters, while if I think of all the individual synapses that make that up, like in connections networks, will be millions of parameters. So this field picture reduces the dimensionality of the parameter space very rapidly, very radically. The same is true for the output. Roughly speaking, for the output would mean that you have some field of activation somewhere, and there is some connectivity pattern that ultimately leads to some motor surface. And motor surfaces in humans would be ultimately ensembles of muscles that drive joints and lead to some motor pattern. And then you could say the regular rule how location of the peak in the of activation or the pattern of activation determines what kind of motor response you get. That gives meaning to this activity pattern. It turns out to be a little bit more complex, because the motor systems always are actually dynamical systems physically. So if you have a motor state, what you really have to look at is the rate of change of the motor state as a function of the motor state. And I will be explaining this at the end of today's lecture in a sort of a simple example. If you think of muscles, it's the reflex loops of muscles that make it so. The reflex loop means that there is some dynamical system that the motor outcome is fed back into the muscle, which adjusts in response to that feedback. So it's actually attractor networks. They have stability, stable states, behavioral dynamics. So I'll come to that. So it's a little bit more complicated. That's why I don't go into more detail now. So that's the notion of fields. Good. A good point to ask any questions you might have. So the next thing I will go to now is to go beyond this picture of just input output and just low pass filtering. And so far the analysis has been trivial, just low pass filtering, just sort of some neurophysical detail. But now we'll see that it really matters. So the activation pattern is a state in the dynamic system sense and matters in an important way. And that will come from recurrent connectivity, which we call often interaction or coupling, that's organized in a particular way, for instance, organized to make decisions, detection decisions, selection decisions, has to do with competition. That's where the dynamical regimes and instabilities come from. You could actually say neural dynamics and dynamic field theory are all about instabilities. So stability is for free, as I just showed you, it's sort of a physical property. Every neuron has stability properties. The membrane has stability properties. But then the connectivity, that recurrent connectivity, makes instabilities, and those are actually really modulating the function of neurons. That's what. So this is the core of the core lecture now, the next unit. So I'll begin in sort of physicist style with the simplest case. It's just one neuron with a neural network with just one connection. It's the simplest possible neural network. So I have a single neuron that self-excites. So self-excitation means that there's a source of input to this neuron that comes from that same neuron, U. It goes through the sigmoid. Sorry, I'm using G and the letter sigma interchangeably. It's awful. In the book, we use the letter G because the letter sigma is used sometimes for widths. And I just can't bring myself to use the G because sigma is much more intuitive. So I'm sorry. Sometimes I use sigma, sometimes I use G. So this is supposed to be the same sigmoid, sigma of U of T. I'm talking about the case where this is a positive number. It's a possibility to look at that as being a negative connection, but I don't want to go into that. The rest still as we had before, so could still receive some external input, right? And again, what would be coming down here would be the sigmoidal activity. In reality, and you will see this in a moment, it's not usually one neuron making a synapse on itself, even though that does exist in the nervous system. It's typically a whole population of neurons that make mutual connections that are excitatory. And then that whole population can be described by effectively having something like self-excitation. I'll illustrate that in a moment. Now that leads to nonlinear dynamics, right? We had the minus U term that makes this dynamics. And then we have the sigmoid added to the rate of change, right? So you see this is a curve that goes to the halfway point and then saturates, right? So what I have to do is add these two curves. You see, here there's nothing from sigma, so here it will be the same function. And then ultimately here is a constant. It will be just shifting a whole minus U term up by that constant. So this here would be just the same as this line shifted up by a constant. And in between, well, it has to transition between those two, so it will do something like that. That's a nonlinear dynamics, right? So far in this picture here, nothing else happened because it's all still has one fixed point, negative slope, and it's all below zero. So everything here would lead to convergence to this fixed point. But if you add to the system now varying inputs, you're just varying the strength of this input here, then different things happen. You see this interesting stuff happening. So I'm using a grayscale. The light gray is very little input, and the dark gray or black here is more input. So across these shadings, input strength varies. We started out here with this case where there's a single attractor here. but you see there are some curves where there is an attractor here, and then there is a fixed point with a positive slope, a repeller, and there is another attractor. That would be a bistable dynamics, and I think I have a slide for that. Yeah, this would be at an intermediate input strength where I have a bistable dynamics, and bistable would mean that if I have some initial state that is here in this right half, right of this repeller, I would be attracted to this on attractor, right? Any of those initial conditions you converge here because it's a positive rate of change here. So go there, negative, go here, right? If you're on the other side of this so-called repeller, then you have a negative rate of change. You would be attracted to this one, right? And here, positive attracted. So it's these solutions here that would all converge to this second, this off attractor. So this is repeller. You see literally because it repels. If you're a little bit to the right of it, you move away in this direction. If a little bit to the left of it, you move away in this direction. So around here, there's some kind of splitting effect, right? Repulsion effect. It's strictly speaking a fixed point. That is, if you're sitting exactly in that fixed point, you would stay there. But if it's just a little noise, a little random input, throw you off that fixed point, and then you go to one of the attractors. I call this one on because if you think of a sigmoid being applied to the activation level, this one would lead to downstream action, this one not. So on, off, some kind of intuitive way of talk about that. So that's a qualitatively different kind of dynamics, right? It's, if you think of the flow, this is a flow that has the splitting point in the middle. It's qualitatively different from this flow where everything converges on one point. Qualitatively different in the sense you can't generate this flow by taking the former flow and just sort of deforming it in some continuous way. Like if it's on a rubber sheet, you could sort of deform that rubber sheet. Here you would have to cut the rubber sheet in one spot. So it's really qualitatively different. That's what the qualitative theory of dynamic consistence actually formalizes mathematically. Now let's look a little bit more into the details of that. So here we had this bistable limit case. We started with a monostable example, and you see ultimately there's a monostable one if I have enough input that has only the on state. So when I increase input, then these this qualitative change is induced. So I'm going from monostable to bistable, and so the interesting part here is somewhere here in the middle. This is where from the bistable, this attractor and that repeller collide to be just tangent and then lift off. That's one of the points where some qualitative change occurs. So if I sort of amplify just this region, then what happens here is this, this would be this repeller solution, the attractor, negative slope, positive slope. If I shift it up by input strength, it will be just tangent here. You can think of the slope at the fixed point as being a measure of stability. And so the slope would become flat and flat. It becomes slow. It takes more and more time to relax. And then ultimately it doesn't relax anymore. And then if you lift off, it's gone. So people sometimes analyze that in terms of a bifurcation diagram where you're looking at the fixed points and their stability as a function of the parameter that induces this instability, see the stimulus strength. And you would see that the unstable branch and the stable branch collide. If you figure that out, they collide like a square root function. And then beyond that, there is no fixed point left here. So this is an example of what is called a tangent bifurcation, the simplest bifurcation of nonlinear dynamical systems. And what it makes it is that this stable state disappears, right? You empirically only see stable states because the unstable states you go away from. So over macroscopic time, you only see the stable states. So you would be sitting in the stable state, moving a little bit, and then this disappears. And what happens then that you go to the next fixed point that exists already out here, which is sort of unrelated to the fixed point we're looking here at here, just following the dynamics. And so you would be going to some other state. We call this the detection instability in the sense that that neuron could be thought of standing for its input. It stands for that function S in a sense. And this would be where the input, the neuron decides to turn on, right, to go into the on state. It was in the off state, so negative activation. And at this point, it would switch into the on state. And that would be a decision that that what it stands for has been detected. And you will see that it plays a very central role in dynamic field theory. This is actually at the core of a lot of phenomena. You will see this makes the sequences and so on. So it's very important. There's a second instability in here, which we call the reverse detection instability, that arises around these two fixed points when I decrease stimulus strength. So if I go from larger to smaller, so then this attractor collides with that repeller at this point, then lifts off. It looks locally like an inverted parabola where these two collide, and again they are tangent. Well, it's also a tangent bifurcation. It's only sort of the stable and unstable have flipped sides, and we call it reverse detection in the sense that if you had already been firing your neurons in the active state, and then it will stop doing that when the input is removed to make this small enough. So let's simulate that again. I wonder if I still have that simulated here. Yeah. So I can simulate that by adding self-excitation. There's a parameter here that allows me to do that. Let's maybe do it a little bit more. I think maybe like this. And you see this is characteristic form. Still, the fixed point is here. Now, if I add input, you see I have the bistable regime. These little markers here show us the fixed point. These are the attractors. That's the repeller. System is still sitting here in this subthreshold case, right here at a stimulus strength, let's say of 2.5. It's quite the symmetrical case. I'm still sitting in the off state, even though the on state is available. And I have to increase, see here the detection stability happens, these two become very close, and then they collide. And just a little bit beyond, whoop, I switch on. Now I have a positive level of activation. If I now go back to this middle, oops, sorry, I played with the wrong parameter. This one's supposed to be 0. I meant this one. If I go back to this 2.5 or something, then I'm staying here in this on state, even though previously I was here in this negative state, the off state. And that illustrates one important thing, that these detection decisions are stabilized. You need less input to keep the decision that you're detecting something than to first induce it. And that's a very general principle that's also used in a lot of applications and probably used by the nervous system in some sense. And so to lose the on state, you need to go to low enough inputs like this, somewhere here, that go through the reverse detection stability. And there it goes through that right away. Yeah. And so if I now give input again, it will stay in the off state. The system, you can actually add noise to this simulation. Noise is just a stochastic input. Like this, for instance, maybe some nice noise here, and maybe that's too much. And you see that the noise actually doesn't do much as long as I'm in the stable regime. But it does a lot when I'm close to the instability. This is where noise leads to a macroscopic change. And so, for instance, let's maybe go to the other case here. Let's stay here, and now I'll really add some bad noise. I can make the system switch just based on the noise. Yeah, and that's a very general idea that you'll also see again in other contexts, that stochastic decisions and random events happen near instabilities. They don't happen when you're far from instabilities. This will be much more dramatic in the case of whole activation fields, where the barrier to a stochastic transition is very high unless you're at an instability. Okay, so what I actually showed you in this little experiment is what is called hysteresis, where here in this simulation is an increase in time of an input, for instance, and then you're going from subthreshold activation, this activation subthreshold, to suprathreshold at the detection instability at a sufficiently high level of input. If you then decrease input, and for the decrease, sort of time is plotted backwards, you decrease so that at the same points you have the same level of input. So you decrease input, then you would switch only at lower levels back to the off state, right? And that will be shown up as hysteresis, that it depends on which direction of change you are going through to see or not see a certain state. And I've actually worked extensively with Hartog to prove that such things really exist in human perception. There have been reports like that in human perception, but they're often tainted by things called response bias or inertia, where people just respond a little later, and you have to distinguish that from actual perceptual state. And there are techniques to minimize other explanations and really provide evidence for this. Now, for the important conceptual importance of the detection and reverse detection instability is that it transforms continuous change into discrete change, continuous in time, for instance. If you here, for instance, I ramped up input in a time continuous fashion, or like a linear function, and then there is this abrupt nonlinear change, which really comes from the instability, right? It's an event. You could say that's the onset event, and this would be the offset event. And that's really the role that detection instability plays in a lot of our architecture. That events, you will see that we'll be doing whole sequences of events, and these events just emerge from the instability. So even if the underlying variables are dynamical, are continuous, they will look like they're discontinuous through instabilities like that. Good. So second step, there will be 3 steps. 1 neuron, 2 neurons, continuously many neurons. So typical physics style, jumping from 1 to 2 to infinite. So when I do 2 neurons, I get a neural network. Now it's already a real neural network. If I were to couple these 2 neurons excitatorily, it would actually behave the same as a single neuron that is self-excitatory. And you will see later that the fields are actually like that. So nothing new happens. So I'm taking the other case where they are coupled inhibitorily. That is, the output, the sigmoided output of one neuron makes an inhibitory connection on the other neuron. So a minus some positive number here. And the activity of the other neuron makes a corresponding negative synapse on the function of the other neuron. Again, this actually stands for 2 small populations, not for individual neurons, and we'll get to that. So one neuron showing up in the equation of the other neuron number 2 of neuron number 1, that's when you get coupling or interaction. These are sort of more physics terms. In connectivity, you would say that's connection, but self-connection versus connection to other neurons is maybe not such a clear concept. So if you use dynamical terms, then these terms are useful, but it's nothing other than connectivity. It's a particular kind of connectivity. So the only way you see that is that other neurons show up in the equation of this neuron. And because this is reciprocal, you can't actually order the neurons so that one is just the input to the other. So this is recurrent, and since there's a recurrent loop, because you could say it goes this way and then that way and this way and that way, right? So the dependency is a loopy. It isn't just, can't be sorted as being just a feed-forward stream. So if U2 was only showing up as input to U1, but U2 itself is not dependent on U1, then it would be still a forward neural network. But because it's a loop, it isn't. Now, this already does a very interesting function. And I'll explain that here in the sort of mental simulation. So I'm looking at these 2 neurons in the simple picture, U1 dot versus U and U2 dot versus U2. That's not quite right, because in every case, I would also have to keep track of the second neuron, right? Because the U1 and U2 are functions of both U1 and U2. Sorry, U1 dot and U2 dot are functions of both these variables. Yeah, there's a request for a break, and I think that's good. I would actually, perhaps it's good to make the break now rather than after this unit. Yeah, let's do the break now.